Staining the gel will
Hello
reveal the
students,
strands of DNA
welcome
on the
back
gel
to the
and allow you to
biotechnology
interpret
principle
your
and
results.
process. So in the previous class we people have seen
This is
the
the
gel
end of lesson 3.
electrophoresis concept. So there I have told you
This
about
resource was
the gel
created at the University of
electrophoresis
Bath
theory
by 2
concept.
natural sciences
So once
students,
again I'll
Ben
tell
Evans
you, I'll
and Ella
repeat the
Baskerville.
concept here. So gel electrophoresis, first definition you have to write. Okay, I have given the assignment for you. What is gel electrophoresis?
This video will take you through the
the
process for
or
loading and
explain the
running
separation and
DNA
isolation of the
samples
DNA
on an
fragments by
agarose
gel
gel.
electrophoresis. So first point is you have to write the definition of the gel electrophoresis.
You
It
will
is
be
a
using a
process
mini
where
subcell to
the
perform agarose
DNA
gel
fragments
electrophoresis.
are
The
separated
subcell
and
has an
isolated
electrode
in the
wire
form of
running across
bands.
the bottom
That
of
is
each
called as
end.
gel electrophoresis.
This provides an
And
electric
here
current to
we are going to
pass,
see
which
that
separates the DNA
this
fragments
gel
in the
electrophoresis
samples.
technique was first identified by the
First, align
scientist
the
called
gel
as
so that the wells are
A.
closest to the
Tiselius.
negative or black
A.
electrode.
Tiselius is the
DNA
name
is
of the
negatively
scientist.
charged and removed from the negative
Tiselius.
electrode through the
Tiselius
gel
is the
toward the
name of the
positive or
scientist who
red
has
electrode.
proposed this gel
Place the
electrophoresis
agarose gel
technique.
into the
Now what
gel
happens
chamber.
in this gel electrophoresis? So third point what you have to write is requirement. In the requirement we are coming to know that
Add electrophoresis running
agarose
buffer to
gel
the
is
reservoirs at
required,
each end of the
plastic
gel
tray is
chamber, and
required,
keep adding
large
buffer until the
comb is
wells in the gel are
required,
covered by at least 2
ethidium
millimeters
bromide
of buffer.
solution is required, then buffer solution is required, switchboard is required, shotgun is required. So all these are the requirements for the procedure. Okay, I have told you also that I'll show
Place
you
your
the video,
samples to be
but
loaded
after
in the
little
correct
revision
order
I'll show you
according
the
to the
video of the
lanes
gel
they are assigned to
electrophoresis.
be run in.
So here
Check
all
your
these are
lab protocol
required.
for this
Now
information.
first gel is prepared. Okay, how the gel is prepared? By
To obtain a
using
DNA
the
sample,
agar powder
slowly
and
depress the plunger on your adjustable
distilled
micropipette
water.
to the
Agar
first
gel
or
is
soft
prepared.
stop.
So agar powder
Holding
from
the
where we
plunger
are
down,
extracting?
insert
From
the tip of the
the
pipette
algae
into
called
the
as
microtube
Gelidium
of the
and the
DNA sample.
Gracilaria.
Place the tip
Gelidium
close to the
and the
bottom of the sample,
Gracilaria
then
are the
slowly
two
release
types
the
of
plunger button.
algae where we can extract the agar
When
powder.
loading the samples,
And
keep the
there
pipette
we
tip
can prepare the
perpendicular to the row
agarose
of
gel by
wells.
adding the agar
This will
powder
reduce the
inside
risk
the
of accidentally
distilled
puncturing
water.
the wells
Then
with
we
the
have to
tip.
cool that
Lower the tip
agarose
of the
gel.
pipette until it
Then
breaks the
we
surface
have
of the
to take the
buffer and is
tray
located just
and
above
fix
or
that
just
tray in the
inside the
buffer
well.
solution. And we have to pour the agarose gel in that. After pouring the cooled agarose gel in that,
Slowly
we
apply
have to
pressure to
create
the
a
plunger
well
button and
by
observe as the
using the
sample
large
fills the
deep
well.
comb.
Make certain you
Then
stop at the first
we
stop
have
on the
to add the
pipette,
DNA
pause, then
samples.
slowly
Okay, we
remove the
have to add
pipette
the
while
DNA
keeping your
samples
thumb
inside
down
the
on the
well.
plunger.
Then the switchboard is on there. We have to on the switchboard.
Once all the samples
As
have been
the
loaded, avoid
current
any bumping or
electricity is
movement of the
passed
gel
through
chamber.
buffer
This might
solution,
result in the sample spilling into
what
adjacent
happens here?
wells.
The DNA
Place the
starts
lid on the gel
traveling.
chamber
DNA
with the terminals
fragment starts
correctly
traveling in
positioned
the
to the
form of
matching
bands.
electrodes on the gel
Okay.
chamber,
Initially, the
black to
larger
black
bands are
and red
going
to
to
red.
cover. Then later on, the smaller bands are going to form. Okay. Then
Connect the electrodes to
afterwards,
the power
these
supply.
bands are
Again,
colorless. I
making
have
certain the
told you.
electrodes match the
As these
terminals on the
bands are
power supply,
colorless,
red to
we
red
have to make them
and
in a
black to
colored
black.
form. So for that, what we will add? We are adding the solution
Switch the
called
power
as
supply
ethidium
on.
bromide. Ethidium bromide solution is added for
Then set
that
the correct constant
particular
voltage for running the
band,
samples.
and
See your lab protocol for
then
this
it is
information.
exposed to the ultraviolet radiation. As it is exposed to the ultraviolet
If a
radiation,
timer
the
is available
colorless
on your power
bands are
supply,
converted into
set the
orange
clock for
colored
the
bands.
proper time for
Okay,
your run.
this is only by only one chemical I am talking. There are several
Press the
chemicals
start
where the
button to begin the
DNA
flow of current that will
bands are
separate the
converted
DNA
into blue
fragments.
color or the green color also.
At this
But
point, you
here
should be
we
able
are
to see
seeing the
bubbles coming
orange
from the
color DNA
wires at
bands.
each end of
Then
the gel
those
box.
bands are separated from the
Notice
gel,
that the
and
positive
that
or red
technique is
electrode
called
has
as
fewer
what?
bubbles
Elution.
than the
What
negative
do we
or
call
black
for that
electrode.
technique? It is called as elution. Now from this we will
In a
get
few
two
minutes, you should
questions.
see the
One
samples
is
begin to
1
migrate
5-mark
from the
question and the
wells
other one
into the
is
gel.
2
Notice as your
1
DNA
marks
samples
question.
run, the
What
dye
is
front
the 1
moves from
5
the
marks
negative
question?
electrode towards the
Explain the
positive electrode.
process of separation and isolation of the DNA fragments by using the gel electrophoresis. That is 1 5-mark question. And the other one is define elution. That is 1 mark question. And the other one is name the chemical component add in the elution gel electrophoresis. That is the 1 mark question. Okay. So now I will show you the video of the gel electrophoresis. Please listen carefully, watch carefully, understand it, and then if you have any doubts, please just while coming the regular classes or doubt clearing classes, in that class you can ask your doubts. Okay, I will show you the video of the gel electrophoresis.
and the fragments will be separated
DNA
based on their
ligation
size.
is the formation of a
Smaller
covalent
fragments will
bond
move
between
faster through
adjacent
the gel
DNA
matrix,
fragments,
and larger
frequently a
fragments will
vector
move
and a gene
slower
of
through the gel
interest.
matrix.
Ligation.
So I'm going to show you how
Welcome
we're
to
going
this
to
video.
set
this
up
is
our
the
gel
video of
box.
the
Intended to
gel
use alongside the
electrophoresis.
third
Mini
lesson in
See
gel
a series
carefully
of
and
results
understand.
on
Try
the
to understand
PCR
these concepts.
process.
Okay.
I have several
This third
items here.
video will
We
guide
have
you in
a
using gel electrophoresis
micropipette
to
for
analyze the
loading our
DNA
samples on the
fragments that
gel,
were amplified
tips for
using
the
PCR
micropipette,
in lesson 2.
a tip waste
For
bucket.
this lesson, you will need
We have
PB92
our
homozygous and
test tube holder
heterozygous control
with
samples.
our samples.
DNA loading dye.
We have agarose.
TAE solution.
And we have our
Note
So
that
this
in
is
your
the buffer
experiment,
solution
this
what
will
they
have
have
been
taken.
pre-prepared
We
at a
have a
1
power
times
supply.
concentration.
Now,
Begin with the
we have
PCR
several different
sample that was
types of power
prepared in
supplies.
lesson
You'll see,
2
you may see a
and
See,
has
different
now
she is
been through the
preparing the gel
PCR
electrophoresis.
process,
I'll show you
resulting
how to
in
use
multiple
this one
copies
today,
of
and
your
That is the
original
shortcut.
DNA
What she is
sample
using now,
fragment.
that is the shortcut.
If they're slightly different, we can work with that in lab.
Take
This
the
is a
loading
DNA
dye
sample.
from the ice
We'll
box.
also need to wear gloves.
So these are
when
called
we're
as
working with the
smaller
gel,
tubes,
because
which
Microliters
the
is
of
gel
the loading
consisting of
contains
dye
DNA
for your
samples.
PCR tube that contains your DNA.
And here we have
So
the
she's
gel
adding
box,
that.
and I'll be showing you how to
DNA
set this
sample
up.
is added.
I'm also going to
Little
wear
DNA sample is
goggles
added.
today because
Discard the
And that
before
syringe
tip
is wasted,
and
then
then
one
gently
more
mix
syringe
it
is
with
taken
bromide,
some
and I
and
don't
of
want
the
once
anything
PCR
to
tube.
splash into my eyes,
Just
so I'm going to wear goggles for today.
add it, sample.
The gel box has a comb.
Next, you
with
will
Sample
need
is
to
ready
create
now.
the
Now she is
agarose
preparing
gel.
agar gel.
Wells in our
This
gel.
may have
So
been done
distilled
for you,
water
and
she is
if so,
taking
you can
and
skip to a
pouring
later
the
point
agar
in the
powder.
video.
Well, comb.
So water
And
and
Take your
you'll
the
1
notice
agar
times
powder in the
TAE
conical
buffer
flask
and
There's a
mix it with
screw,
some agar
and
powder.
these wells are adjustable, and that will come up
In this
later.
example, 2 grams
This
of agar
is
has
a
been
little
used
spacer.
with
It
200
has a
milliliters
thick
of
side.
TAE 1 times buffer solution.
A thick side and a thin side, and I'll show you what that is for later as well.
To prepare the agarose gel, this mixture will
When
need
we
to be
pour
boiled.
the gel, we have a
Therefore, in
gel
order to minimize
tray,
evaporation of TAE
and
during
we
this
have a set of
process,
rubber
wrap
dams.
cling film over the top of the conical flask twice and pierce with a sharp object.
This is our lid, and the lid is connected to the electrodes, which will be attached to our power supply.
The mixture is now
And this
ready
is
to be
safety
placed into
lid,
a microwave.
so no power will go
Put
to
it
the
in the
unit
microwave for
unless
1
the
minute,
lid
and at
is
the end of the
secure
minute, check to see
on
whether
the
the
gel
mixture
box.
has become transparent.
This is our gel box, and if you notice, there is a leveler in the center of the box,
If it is
and
not,
that's the
then
first
put
thing
it back
we
in the
need to do.
microwave
This is called
for
as
another
agar
minute.
agarose gel.
Continue
Level our
this
gel
process
box
until
so
it
that when
has.
we pour the gel will have a
Note that the
flat
conical flask can get hot
surface.
during this process, and something
So
may be needed to
You'll need
protect your
to
hands from
adjust
the
the
heat.
screws in the legs.
When the agarose mixture seems
of
to be
the gel
transparent,
box
it may be
until the
useful to
bubble
hold the mixture up to a light to double check this.
is in the center
The mixture needs to be fully transparent before moving on to the next stage.
of the circle of the leveler.
When this is complete, you will need to create the
Now she
Once
is
agarose
you've
gel
finished
preparing for the
adjusting
casting
gel
tray.
gel box,
electrophoresis tray. You can
Assemble
see
the
bubble is
casting
in the
tray,
how she is
center
making sure
fixing
that
the
the
tray. You
rubber
can see
seals
You're
on
ready
it
to
are
pour your
tight.
gel. So
that is
let's
a
put
tray, gel slab.
the
What
gel
do we
tray
say?
together.
Gel
Notice that
slab.
the
it
rubber
is.
dams have a
When
thick
the
end
casting
and
tray
a
is
thin end.
assembled,
We want
gel
to
take the
slab.
sample
put the
cone
dam
and place it
onto
across
the
the
gel
tray
tray
within
with
2
the thick
centimeters
end
from the end.
at the top.
If
So
you
we
want
just
to
push
increase the
large
number of
teeth
samples
combs, you
that
can
are run
see.
on one
onto the
gel,
tray.
you can
She
also
red
place
color
the
one,
sample
large
cone
teeth
in the
comb
middle
it
of the
Do the
tray
same with the
to
other
create
side,
more
again
wells.
making sure that the
Your
thick
casting
end
tray is now ready to have the
She's
agarose
pouring the
gel
elec-
mixture
gel-
poured into it.
Cooled gel.
Note
Now
that
you're
the
all set with
agarose
your
needs
tray.
to have cooled to
The tray
60 degrees
sits
Celsius
down
before this can
into
happen.
the gel box and
Leave the gel
should
to
now be
cool
level,
and
and you're ready
solidify for
to
10 to
pour the
20
gel.
minutes.
So
It will be
they are told to
translucent
cool it.
when ready to use.
On
When
the
the
gel
agarose
tray,
gel has set, remove the
you
sample
should be
cone
able
from
to
the
see
see
gel.
how
a
large
little
teeth comb
line,
is
and
removed
that
there.
is the
See
pour
the well.
line. You don't want to pour
This
the
should have
gel
created
any
wells in the
thicker
gel
than that.
where your DNA
So
sample
you want
can
to
be
Wells are
make
placed
sure that
later
when you
on.
pour your agarose into the tray, it's
Yes.
below the
The
gel
next
line.
stage of the experiment
That is
will
a switch
use the
port.
electrophoresis
So
There
equipment.
she will pour the buffer
and
solution.
because we're using ethidium bromide, we're going to wear
Remove
gloves.
the rubber sides from the casting
Ethidium
tray
bromide
containing your
is a
agarose
potential
gel and place
mutagen,
it onto the platform in
and
the
we
gel
put
box,
it into the gel.
making
It's
sure that
used
the
as
sample
a
wells
stain.
are at
It
the
will
cathode or
bind
black
to the
end of the
DNA,
base.
and when we're done, on, we can view the DNA using ultraviolet light, and we'll
Next,
be able to see
pour
the DNA on
approximately
our gel.
275 milliliters of
Once
your
you've got your
TAE
gel box
buffer
set up and you're ready
into
to
the
pour, you want to
electrophoresis chamber
check
until
out
it
your
just covers
comb.
the
And
wells
this
in the
comb,
gel.
we're going to use the 8-well side of the comb. We'll form
The
the
gel
wells
is now
in
ready
the
to
gel.
start having
But
DNA
before we
samples
pour the
loaded
gel, we
into
just
it.
want to make sure that there's a space between the bottom
The
of the
control
tray
DNA samples
and
will
the
first be
comb. You
loaded
don't want the
into the
comb to go
wells.
all the way
Make
through
sure
the
that the
gel, or you'll
pipette
make a
is set to
hole, so
10
when you add your
microliters when
sample, the
putting in
sample
the
will go
control
right through the
DNA
gel.
samples.
You want the sample to be
To ensure that there is no
in the
cross-contamination,
gel and move through the
discard
gel
the
matrix.
pipette tip
So
and
we're
swap
going
it for a
to
new
put
one
this
between every
into
sample.
our gel box, like so. And then we're going to take the spacer and use the spacer to make sure the spacer
She's taking
fits
the new
underneath
syringes and she's
our
adding the
comb.
DNA samples over there.
Be sure to add
You can
each
see
If the
DNA
spacer
sample
adding
fits
the
to a
DNA
new well
samples.
every time,
then you know
so that
that
the different samples do
your
not get
comb
mixed.
is set properly and you're going to make wells. You're not going to make holes in your gel. Now you can remove the spacer.
Continue to add 10 microliters of
and
each control DNA
remove
sample and then 20 microliters
comb
of each
and you're
student
ready to
DNA
pour your
sample
gel.
to the wells in the gel.
The other thing we should check on is the wells have to be at the negative electrode end of the gel.
Continuously she's
When
adding
every
the
DNA
DNA
sample has
samples.
been
From the
added,
wells
place the lid on the gel box
to
and
the
attach
positive
the
She's
lid
connecting
to the
that
electrode
to
power
the
other end.
supply.
switchboard. See the
The lid
connection?
will only attach to the base in
We're
one
going to just
orientation,
put the lid on and make
red to
sure
red
that we
and
have
black to
the
black.
we're going to put, when we put our comb in, it's going to be at the negative end. The black electrode here is at the negative end, and the red electrode is at the positive end, and there's only one way to put the lid on, so you can't
Set
make
the
a
power
mistake.
supply to
So
100
we're at
volts
in the
and
right
let the
orientation.
electrophoresis run
Our
for 30
wells
minutes.
are at the negative
You should see
electrode.
bubbles moving inside the gel
now we're
box
ready
as a
to
result
pour
of the
our
current
augros.
being passed through. During the electrophoresis process,
She's showing
your
the
DNA
mechanism
samples
order.
will separate
go
along the
above
agarose
the
gel
fill
based
line.
on their size. Shorter strands of DNA will move along the gel more than the longer ones.
Place the
This results in
comb, the
different
8-well
amounts of
comb,
separation for different samples of
at
DNA.
the negative
When the
electrode
electrophoresis
end
is
of your
complete,
gel. And you
turn off
want
the
to
power
make
supply and
sure
remove the lid from the gel
that
box.
the comb
Carefully remove
is
the gel
not
tray containing the
jammed
gel
up
from
against
the gel
rubber
box.
dam like this.
Take
you have
care
to have
when
a
removing the
space
gel tray,
between the rubber
as the
dam
gel will be slippery.
and the
Using your
comb.
thumb, nudge the gel from the
Now
gel
we're
tray,
just going to let
allowing
that
it to
sit
slide
for about
into your
20
plastic
minutes
staining tray.
until the gel hardens.
Your gel
And
is
as
now
the
ready to be
agarose
stained.
hardens,
You can
it will
see the
become
bands
Staining
over
opaque.
here.
gel will
Just
reveal
you
the
just want to
strands of
make
DNA
sure
on the
that
gel
it is
and allow
hard
you to
before you remove
interpret
the
your
comb,
results.
otherwise the wells will collapse. While
This
you're
is the end of
waiting
lesson
for
3.
your gel to harden, you have
Well,
about 20
that
minutes.
was the gel
This resource was
electrophoresis.
created at the
Okay.
Make a
University
gel
of
map
Now-
for your samples as described in
So
your
hope
lab
so you have
manual.
understood the gel electrophoresis
And
technique.
you can also prepare your
If
samples in the sample
you
buffer.
can rewind if you are
sample
not
buffer
understood, just
contains
rewind
a
and see
dye, a
the
tracking
gel
dye,
electrophoresis technique.
which will
Now next
allow you
we'll
to
go
track
for
the
the
DNA
vectors.
as it moves through the gel
Next we are
matrix.
going for the vectors.
And it also contains
So first we have
glycerol, which will make the
completed
sample
the
heavy. So
first
that when we
concept,
add the
first tool
samples
of
to the
genetic
wells
engineering,
in the
that is
gel, the
enzyme. We have completed that.
samples will sink to the
Then
bottom
we
of the
people have
well
seen the
underneath
concept
the
called as gel
aqueous buffer.
electrophoresis. Now the
While we're waiting
second tool
for
of genetic
the
engineering, what we are going to
gel
see
to
is
harden,
the
can set up your
cloning
samples.
vectors.
And just
Now
like in the
students,
cooking
vectors
shows, we
are
have
what?
an already
Vectors are nothing
solidified
but
gel.
these are the
And as you
vehicles
can
of DNA.
see,
What are
the
vectors here?
gel is
Vehicles
opaque
of DNA.
and it has
So
a
these,
solidified. This is a
why they are
2%
called as
agarose
vehicles of
gel,
DNA?
and this
Because
gel acts as a
they
matrix through which
carry
the
the
DNA will
DNA
migrate and will be
from
separated
one
by
generation to
size.
the another generation. That
We're
is a
gonna
normal one.
carefully
But
lift
here from one organism to the another
comb
organism we are
out
going to see.
of
The vectors are
the
called
gel.
as
Place that
vehicles of
aside.
DNA because they are carrying the DNA from one organism to the another organism. So here we are going to see
And
so many
then
types
we
of
will
vectors.
carefully
Okay, we are going to see
remove
so
the
many
rubber
types of
dams. Now
vectors.
sometimes you
For
have a little bit of
example,
suction
plasmids
between
are there.
the agarose
Cosmids
gel
are
and the
there,
rubber dam, and you want to be very careful.
bacteriophages
You
are
don't want to
there,
break
5
the
virus
gel.
is there,
So you can
plasmids are there,
carefully
PHAS,
try to
plasmids.
remove the
So all
suction,
these are the examples of the
release the
vectors.
suction between the gel
All
and the
these
dam.
are what?
Another
They are the
trick
examples
is
of
to use the little
vectors.
spacer, the
So
thin side
here
of the
in your
spacer,
syllabus they have mentioned
and
about
just
the plasmids.
put it down in between
What they have
to
mentioned?
release that
They have mentioned
suction
about the
between the gel and the
plasmids.
dam
Okay,
if necessary.
plasmids and bacteriophages
Now, once
have the
these are off,
ability
you just
to
want to make sure that you
replicate
don't
within
tip this
themselves.
either way, or your
First
gel
of all,
could
you have
slide
to
right off
understand
this gel
what
tray.
are plasmids and what are
So we'll
bacteriophages,
place
that you
this
have to
back
understand.
into the gel box.
So here students, plasmids are double-stranded,
And now we're ready to add
self-replicating,
our running buffer.
extra-chromosomal,
So take your
circular
solution of running buffer
DNA.
and pour it into the
What
gel
are plasmids?
box,
1 mark question it is.
completely covering
What are
your
plasmids
gel
here?
and making
Plasmids
sure
are
you have
double
buffer in
stranded,
both of the
self-replicating,
reservoirs.
extra-chromosomal, circular,
The other thing you want to
DNA.
make sure of
That is
is
the
that your
plasmid.
wells, which
And
are going to contain your
bacteriophages, you people know
samples,
very well. What are bacteriophages?
are
The
completely
virus
covered. You don't want to see any
infecting the
pucker
bacteria
marks
is called as bacteriophages. What are bacteriophages? The
over the
virus
wells
which
where
are
they're
infecting
not
the
completely filled
bacteria
with
are
fluid.
called as bacteriophages.
So now you're ready
So
to load your
plasmids and
sample.
bacteriophages are the most common vectors used in the biotechnology. Okay, plasmids and the bacteriophages are the most
And
common
again, before
vectors
you
used
load
in
your
the
sample, there are a couple of
biotechnology.
things you want to make
Once again, I
sure
will
that you
repeat.
do.
Vector is
Once
the
the
second
sample
tool
is
of
loaded, you
biotechnology,
don't want to have to
where
move the
we
gel
are going
box
to see
around
that
because you
these
could slosh
vectors
the
are called
samples
as
and they could
vehicles
slosh
of
out of the wells.
DNA.
You don't want to do
What
that.
these vectors
So
are
make
called?
sure
These
that
vectors are
wherever
called as
you
vehicle of DNA.
finally
Why? Because
load
they
the
carrying
samples,
the
that
they
you
are
are
carrying
close
the
enough to your
DNA
power
from one
supply
organism to the
so that
another
you
organism.
can attach
And
the
the most
lid,
common used
the
vector
electrodes on the
is
lid to the
plasmid
power
and the
supply. You won't have to move
bacteriophages.
the gel box.
What are plasmids
Also
here,
make sure
students?
the gel box
Plasmids are the
is in a safe
self-replicating,
place where it
double-stranded,
won't get
circular
knocked off
DNA.
the bench.
That is a 1 mark question. Bacteriophages
Again,
means
check
what?
to make sure
Bacteriophages
that your
means
wells
the
are
virus
at the
infecting the
negative
bacteria
electrode
are
because
called
DNA
as
is negative and will travel to the
bacteriophages.
positive electrode.
Now, for
Because
your
again, you do
syllabus,
not
they
want
have
to have to
mentioned the
move
structure
this
called
around
as
once your samples are in the
PBR322.
wells.
Okay, you can see this diagram.
While the gel
I'll
was
show you
hardening,
the
you
diagram
prepared
of
your
the
samples.
structure. Yes, this is the
And
structure
you have a
of
standard and you have a set of
PBR322
samples.
as
And
a
I'm only going to
plasmid for
add
your
2
syllabus.
samples to the
Most
gel as a
common
demonstration,
used
but
plasmid
you
it is.
each have
That
2
is
samples, and there are 3
PBR322.
of you, so you'll have 6
Okay,
samples
so
plus
here you can see
your
the selectable markers
DNA
are
ladder
there.
standard.
Okay, then multiple cloning sites are there, then ORI site are there. Okay, selectable markers means here you can see ampicillin resistant gene and
Each of your
tetracycline resistant
samples
gene
should
are
be
the
about 10
two
microliters.
types
You
of
should have about
selectable
10
markers.
microliters of sample
Then
left
origin of replication site you can
in
see, and other
your
these are
microphage
all
tube.
called as
If
multiple
you don't have
cloning
10
sites.
microliters, you just
I'll
have
show
to
you
use
the
whatever you have
video
left.
of the vector also. I'll show you the video of the vector
So you're
also.
going to pipette
But
this out
first you try to understand what is this cloning vector. Okay.
into your
So
pipette tip,
here
and
you
you
can
want to
see
avoid
plasmid
getting
and
bubbles.
bacteriophages have the ability to
And then
replicate within bacterial cell
sometimes it's a good
independent
idea
of
to
the
sort of
control of
guide the
chromosomal
tip
DNA.
down
See,
into the
plasmids
wells
are
while you're
also
loading.
self-replicating
So you have to
process.
get into a position so that you can see
Bacteriophages
the
are also
wells.
self-replicating
Carefully
process. These
put the pipette
plasmids
tip
where they are found
beneath
inside
the
the
surface of the
bacteria.
liquid
That is why except
and
the
depress
bacterial
the
DNA,
plunger and go
except
down to
the
the second
bacterial
stop
DNA,
and
they
pull up. Now you
also
may
have
see
the
a little bit of
capacity
dye,
to
that's
multiply
okay.
themselves.
Don't
Okay,
worry
they
about
also
it.
have
But
the
you want to
capacity
get
to
the majority of your
multiply
sample
themselves.
into
Those are called as plasmids. If you remember the
well.
PUC first year
Do this
class,
one
there
more
I have
time.
shown you the
Second
structure
sample.
of bacteria. In that structure of bacteria, you people have
Again,
seen the
pipette up
structure
your
of
sample.
plasmids. Okay. And also the bacteriophages also have the responsibility of multiplying themselves. Okay,
Put the
they can
tip
produce so many copies
just
of
into the well.
bacteriophages, and they can
load it.
increase
Now
their
in
genome in the
lab, we're
bacterial
going
cell.
to have some practice
Bacteriophages
gels
itself, they are
set
growing
up
in
so
the
that you'll be able
bacterial
to
cell within
practice
a million,
loading if you would like
within
to before you
20
actually
minutes.
have
Within
to do
20
it on your
minutes,
gel.
a million
A couple of
fractions
things
of
to be aware of is that you do
million
not
number of
want to
bacteriophages
poke the
can be grown
pipette
within
tip
20 minutes. Like that
down
replication
into the well, or
capacity
you'll
the
poke a
bacteriophages
hole in the
they have.
gel, and then your sample
Suppose
is
in
not gonna work.
case any
Once
foreign
you have
DNA
loaded your
particle,
samples
suppose
based
my
on the
DNA
map that you
is
made while
attached
you
to
were
the
waiting for
plasmid
the
DNA,
gel to harden,
then they can be
you're
multiplied
ready
into
to
number of
attach
copies.
the lid
Okay, then
and
they are
hook
multiplied
it
into
up to the
number
power
of
supply.
copies. That is,
Notice that the lid
these,
has 2 little
since
clips
they have the
on
capacity
either
of
side.
self-replication,
Those need to
since they
slide
have the
into
capacity of
little
self-replication,
knobs
they
on either
can
side of the
multiply
gel
in
box.
any of the number.
So place
Okay,
the
that is
lid on
about the
top,
plasmids
slide it
and the
forward,
bacteriophages. Now students, vectors
and
nowadays
make
it is
sure
very common in the
that it
biotechnology.
is secure.
Why? Because
Make sure
without
that you
the
don't have any
vectors
leaks.
there is no products synthesized in the biotechnology. In whichever area you take, in the field of medicine it may be, in the field of
And then
animal
we're going to
husbandry, in
hook
the
up our
field of
electrodes. It's
plant
very
cultivation,
simple.
wherever you
Red
take,
goes to
without
red,
the vectors no
black to black.
field is there where the vectors are not utilized here. Okay. To
And
insert the foreign DNA molecule, the vector is
what-
required. It is
What
must
you
and
want to
should.
do before
Now
you
we will see
actually
what are the
plug in the
characteristic
electrodes is
features
make
of the
sure
vector.
that
Okay,
the
what are the characteristic
power
features of the
supply
vector we
is
are going to
turned
see.
off.
Hope so you
this
have
was turned
understood
off
this one.
before I put the
Hope so you have
plugs in,
understood this
because we're
one.
working with
That is
electrical
cloning
source,
vectors we
don't
are going
wanna
to
have
see.
any
So vectors are
problems.
what here? Vectors
And you'll
are the
notice
vehicles
that
of the
there's a caution
DNA
on the side
where
of this,
they are carrying the
that you're working
DNA
with
from one organism
electricity.
to the another
So we will now
organism.
turn on the gel box.
Most
gel
commonly,
power
so many types
supply.
of vectors are there, but we are using the plasmids
And
and
we're
the
going to
bacteriophages
set
as a
it
vector.
to
What are
about
plasmids here?
100 to
Plasmids are
105
the
volts.
double-stranded, self-replicating, extra-chromosomal circular DNA
Looks
where
like it's
they are
right about where we
present
want
inside
it,
the
but this is
bacterial
the
cell.
adjustment knob if
And
you
the
need to adjust
bacteriophages
it.
are those DNA
If it's
fragments
not exactly
where we are going to
100
see
volts,
the
that's okay. We want it to be somewhere,
what we are going to see is how
around
they are
that
going
voltage.
to
Notice that
multiply
this
themselves within a fraction of second
switch
in
adjusts
the
for milliamps
bacterial
and volts,
cell.
and you want to
Now
make
we'll
sure
see
that
the
it's set on
characteristic
volts so that
features
what
of
you're
the
reading in
vectors here,
the
plasmid or the
window
vectors
is
here.
volts, not
So as
amps.
I've already told you, that
So
is
now
origin
our
of
gel is
replication
set. We
site.
have power going to the gel
ORI site or origin of replication
box,
site says
and our
that
samples should begin
it is the
to
sequence
migrate.
where replication starts. What is
One
the
thing you
ORI
should
site
look
or the origin
for
of replication
before
site?
you walk
It is the
away from your
sequence
gel.
where
This
the
is going to take about
replication
30 to
starts.
45 minutes.
And
if your
in
professor wants you to, you could actually possibly increase
from this
the
site
voltage
only,
and do it a little
so
bit
many
faster,
copies
but,
of the recombinant
the lab
DNA
manual
are
says 105
synthesized.
volts, which takes
Suppose
about
any
45 to
foreign
50 minutes.
particle. See, if I show you the
But
diagram,
before you leave
you
your
can see the
gel,
diagram
a
here.
couple of things you can do is check to see whether or not the
So
wires
this diagram you can
in the
see.
gel box have
So
bubbles.
this diagram, here you
And
can
if
see the
you see bubbles
ORI
around the
site
wires,
or
you
ORI
know there's
site,
electrical
or
current
you can say
flowing.
origin
If you
of
don't see any
replication
bubbles,
site.
then there's something
So
wrong
this
with
is
the
the
gel box
part
and there's no
that
electrical
is
current flowing.
TETR apostrophe
Sometimes we
R.
have problems
It is
with
the
the lid,
part
so you
where
really
the
need to
foreign
check
DNA
that.
is inserted.
The other
When
thing
the
you want
foreign
to
DNA
do
is
is make
inserted
sure that
here,
your samples are actually
it is
migrating
joined
in
by
the
the
proper
ligase.
direction.
It is cutted. This portion is
Once you
cutted by
see
the
that, then
REN
it's okay
and it
to
is
go
joined
away
by
and
the
go
ligase
off to the
enzyme.
retreat and have
So
something
here
to
what
drink, but I
happens
wouldn't leave for more
with
than 30
this,
or 40 minutes.
after coming and joining
thanks,
the
and I'll
foreign
see you in
DNA
lab.
here, it will become recombinant DNA. That recombinant DNA is multiplied. Okay, that recombinant DNA is multiplied as usual, like how the normal DNA is undergoing replication process. Okay, so it is the origin of replication site where it is going to help for the multiplication of the plasmid. Okay, for the replication of the self-replication of the plasmid. Now students, like this, per cell, that means one cell in one bacterial cell, there is a presence of 20 to 30 types of plasmids. That means 20 to 30 number of plasmids. How it is synthesized? By the origin of replication site. Okay, by the origin of replication site, what happens? There is an arising of 20 to 30 number of plasmids within one bacterial cell. That process is called as cloning. What is the meaning of the cloning? If you remember the first topic, reproduction organism, there the madam has told that cloning is nothing but the offspring which is genetically and morphologically similar to that of parental cell. So imagine that the one your own DNA is shifted here, okay, by using the RNA and the ligase. Now this will undergo self-replication. So with the help of the ORI site, with the help of the origin of replication site, we are going to see that there is a production of the 20 to 30 cells per bacterial cell. Okay, so that is the function of the origin of replication site. Okay, once again I'll repeat, the first part of the plasmid is origin of replication. Origin of replication site is nothing but the point where the replication starts. What is ORI site or the origin of replication? That is the point where the replication starts. That is called as origin of replication. This is going to help for the self-replication, and here we are going to see that this particular place, that is origin of replication site place, is helping for the production of 20 to 30 plasmids per
Now,
bacterial cell. That
what
much
we
of
do is that
capacity
we
is
take
it is
this
having.
solution
Okay.
and we pour
That
it
we
in
are
a plastic
completing about the
container
origin of
like this,
application now.
and we
Next
add
part what we
this
are going to see
plastic
is
piece in order
selectable
to
markers. You
create
can
some
see here.
wells
You
in
can go for
the
the next
gel where
part,
we apply our
selectable
samples.
marker.
The
You
gel
have seen the
takes
origin of replication
20 to
site.
30 minutes
Okay.
to
So this is the
solidify.
origin of
Once the gel
replication
is
site you have seen.
solidified, we put
Now
this
next
plastic
is
container
selectable
in,
marker. So
in a
students,
chamber,
selectable
and we
markers
fill it
are
with the
those
same buffer.
where
First,
there
please
is
pour
an
it
insertion of the
80
foreign
buffer, the
DNA
same buffer we
particle.
used
See, I'll show
in
you
the
the
gel.
diagram once again.
And then we remove
Ampicillin
this
resistant gene,
plastic piece, so we end
tetracycline
up with a
resistant
gel
gene.
looking like
These
this.
are the two
So this is the
selectable
gel from
markers.
above. This is
Okay,
our gel.
these are the
These
two
are the
selectable
wells when we
markers
apply
which is there in
our
this
DNA or
plasmid.
RNA samples.
Whereas in the another
What we do
plasmids
then
and
is
all, we
that
will
we
see
apply our
another type
DNA
of
or RNA
selectable
samples
markers
in these
like
wells, and
kanamycin,
then we apply an
chloramphenicol.
electrical
All these
current
are the
using
names
a
of the
battery or a
selectable
power
markers.
source,
What is the use of the
a
selectable
negative
marker?
charge
That
toward the
is,
samples
they
and a
are
positive charge
allowing
on the other side of the
to
gel.
grow
What's going
only
to
the
happen is
recombinant
that the
plasmid.
nucleic acids are
They are
going to
allowing to
migrate
grow
from the
only the
negative toward the
recombinant
positive charge.
plasmid.
Why? Because
Not
the
that
nucleic
non-recombinant
acids are
plasmid.
negatively
Non-recombinant
charged.
plasmid means
Why
what?
the nucleic
The
acids are
plasmid which do
negatively
not have
charged?
the foreign DNA.
Because of this.
Non-transformant we
Because of
will
the
say. The
phosphate.
plasmid
So we know
which
that
do not have the
nucleic
foreign DNA,
acids,
that is
either
called as
DNA or
non-transformant
RNA,
plasmid.
are composed
Transformant
of
plasmid
sugar,
means
either
the plasmid which is
ribose or
consisting of the
deoxyribose,
foreign DNA.
and the
The
nitrogen
plasmid
base
which is consisting of the
and
foreign
phosphate
DNA is
groups.
called as
So the
what?
phosphate are
Transformant
negatively
plasmid.
charged, and because
So
of this,
these
any
selectable
sample of nucleic
markers,
acids, either
one
DNA or
selectable marker
RNA,
is not
is negatively
allowing
charged.
to
And
grow
because
the
of this, the
non-transformant
nucleic
bacteria.
acid samples
Another
will migrate
selectable
from the
marker
negative
is
toward the positive charge.
taking the foreign DNA into its
Now,
part.
what we should do is that
For
first of all, we
example,
should prepare
what
our
diagram you
sample.
are going to
The
see now? Here
sample
two
of
selectable
DNA or
markers are there.
RNA is
One is
extracted
called
from a
as
cell.
ampicillin
So
resistant
either
gene,
we
other
extract
one
the
is called as
DNA or the
tetracycline
RNA.
resistant gene.
Then we mix the
Ampicillin
sample with
resistant
loading
gene
buffer and with the
is the
restriction
type of
enzymes.
selectable
I'm going to speak
marker
about the two of them.
where it
First,
is
the restriction
allowing to
enzymes
grow the
are very
transformant
specific enzymes. They
bacteria,
are in the
or
nucleases,
it is
so
allowing
they
to grow the
cut the DNA.
recombinant plasmid.
And this thing is that why
Tetracycline
they are
resistant
called
gene is
restriction,
the
because
one
they
type
cut
of
the DNA
selectable
in a very, very
marker
specific
where the
place.
foreign
So
DNA is
there are
inserted.
many restriction
What is
enzymes,
inserted?
but each one
Foreign
of them
DNA is
can
inserted. Once
detect a
again, I'll
specific
repeat.
sequence of the
Selectable
DNA,
markers
specific sequence
are
of
those
nucleic
markers which play an very
of
important
nucleotides.
or
So
vital
let's say
role in
one
the
restriction
plasmid.
enzyme can
In the
detect the sequence of
PBR322, you are
CCGT,
going
for
to see two
example,
types of
and then once
selectable
this
markers.
enzyme
One is
see
called as
the
ampicillin resistant
CCGT,
gene
it cuts
and the other one is called as
downstream the sequence.
tetracycline
So
resistant
each
gene.
restriction enzyme can
Ampicillin,
detect a specific sequence and cut
AMPICILLIN.
downstream
Ampicillin resistant
downstream this
gene
sequence. What we're
is
going to
the
have then
selectable
is smaller
marker
pieces of
where
DNA.
it
Why do
is
we apply
not
restriction enzyme?
allowing
Is
to
because the
grow the
DNA is a very long chain, so we
non-transformant
cannot apply
bacteria
it
or
in the gel, but we need to
non-recombinant
cut it
bacteria.
first.
It is not allowing to grow that one.
The loading buffer
Whereas
is composed of
tetracycline resistant
glycerol
gene
and
is the
coloring dye.
place where the
First,
foreign DNA
the
is
glycerol
inserted.
is used
When
to
the foreign
increase the
DNA is inserted
viscosity
into
and
this
the
DNA,
density of the sample.
both the DNA will
Why?
combine and
Because
it is
once
going
we
to form the
apply the sample
recombinant
in the
DNA.
well, we want the
Are you getting
sample
my
to
point?
settle down.
When the foreign
Otherwise, it will
DNA
be
is
mixed with
inserted into
the
this
buffer, and then we
plasmid,
will lose
both
our
the
sample.
DNA are
But to
going to
increase the
combine
viscosity,
and they
when you
are
increase
going
the
to form the
viscosity
recombinant
and then
plasmid.
the density
Understood?
of
So apart
the
from
sample
the
will
two
settle down
selectable
in the
markers, you can
well
also
and will not
see
be
the
mixed
another
with the
selectable
buffer.
markers also.
The coloring
You
dye
can also see
mostly
the
used is
another
bromophenols
selectable
blue.
markers also like
It's a blue dye.
ampicillin,
It's used to give
chloramphenicol,
the sample a color,
kanamycin,
so we can
etc.
detect the
Okay,
sample or the gel
chloramphenicol,
once it
kanamycin, they
migrates.
are seen in
We
the
can
another
see
types
that the
of
sample, for
plasmids.
example,
Okay,
reaches the end of the
chloramphenicol and
gel,
the
and then
kanamycin, they are seen in the
we turn
another
off the
types of
power source.
plasmids, whereas the
Now,
ampicillin
how
and
does
the
it work?
tetracycline resistant gene
Let's say
is seen in
that this is
the
our
your
gel. The well will look
PBR322
like
structure.
this from
Is
a
seen in your
side look. So what
PBR322
we do is that
structure.
we
Hope so
apply
you have
our
understood these
sample
two
in the
parts.
well.
First of all,
So
first
it will
part
look
is
like this.
origin of
And then
replication.
we
What the
apply
origin of
the
replication
electrical current.
site says?
So the
It
sample
is
will
the
start to
point where
migrate
the
from the negative toward
replication
the positive charge.
starts
The thing is that
and
the
per cell there is an arising of 20 to 30 cells. Whereas next what you have seen is selectable markers. So in the
smaller sequence will migrate
PBR322, two
farther,
types
and the
of selectable
smallest
markers are
sequences
the
will migrate
ampicillin
the
and
farthest.
the tetracycline. So
Uh,
ampicillin is not allowing to grow the
Now you can imagine it
non-recombinant
this way.
DNA,
So let's say
whereas
that this is a
tetracycline
gel. There
is
are pores
consisting
inside. As I told you, the
consisting
pore
of the
size
foreign
depends on
DNA.
the
So
concentration
with this
of
class,
the
we'll wind up that
agarose
next
buffer
part we'll
solution.
see in the next class,
So
and
yeah,
also I'll show you
larger
the video
sequences
of
cannot
the
migrate farther,
vector
but the
first,
smaller
then I'll do
sequences
the
can
explanation.
migrate farther.
Thank you, students.
Now,